Okay, good afternoon, guys. We're going to get started here on this Wednesday. I hope everyone has had a good start to their week. We're going to keep going here with our discussion of renal physiology. And as a reminder, what is ahead of you? You've got another set of lab data that's due tomorrow night, lab 11, week 2 renal. It's the same sort of math that you did for the first week. So if you got points out or you got some things wrong that first week, please make sure you look at the comments so you can do it correctly for tomorrow. And then you've got one more lab after that. Also due today or this week, you've got a few activities, and then next week, next Monday, you've got that paper. So you've got some things to be working on. And as always, I'll try to keep this to around 25 minutes or so. And the chat is open if you have questions. See the chat. Okay, so where did we leave off? We left off, we're talking about the nephrons. We're focusing on this functional unit of the kidney, and we went through the tubule part, the part that transports that filtrate, and we talked about what each segment does. We went into the cells there. Remember, the tubule is lined with one cell-thick wall of epithelial tissue, and we talked about what kinds of transporters are in each part and what they do to make the filtrate eventually into the urine, and have it have the correct concentrations of ions and water and all that good stuff. Okay, so this is the question to consider that we had left off with last time. So how does the kidney maintain glomerular filtration rate as blood pressure changes? So we talked about how we can think about the GFR and the glomerulus in general as the metaphor of a hose attached to your house. So you have a house, you have a tap coming out of the house, it's got a hose. In the middle of the hose, there are some holes that's letting some water go through, and then there's more hose after it. So the tap attached to your house, that's like the heart that's sending the water, or in this case blood, into the first part of the hose, the part approaching the holy part, which is our afferent arterial. Then we've got the holy part, our glomerulus, letting a bunch of stuff go through. And then we've got the part at the end, the efferent arterial. And we want to maintain the same rate of stuff flowing through that filter. We want to maintain our GFR of plasma mils per minute moving through that filter. How do we do that if our blood pressure is changing? If we're changing the amount of force leading into that filter? Well, there are both intrinsic and extrinsic regulators that control GFR. Intrinsic, things that happen in the kidney, and then extrinsic are hormones acting on it released by other organs. And the goal here is to maintain a constant GFR over a changing blood pressure. Blood pressure goes up, we want to make sure the GFR stays the same. Blood pressure goes down, we want to keep the GFR the same. What can we do in the kidney to do that? And there are three ways we're going to talk about first with this intrinsic control. So we have myogenic regulation, tubular-glomerular feedback, and mesangial control. Okay, so we have three ways we're going to start with. And overall, we're looking at what's happening in part to some of the cells around the glomerulus, and they're changing in response to blood pressure. And keep in mind, if unregulated, high blood pressure would increase your GFR. So if you increase the amount of stuff, of force into the filter, you're going to increase the rate at which things go through the filter. Which we don't want to do. We want to make that constant. So the first is myogenic regulation. This is where we're going to increase the arterial pressure in our afferent arterioles to decrease our GFR. So as afferent arterial pressure goes up, it's going to activate stretch receptors in the walls of that arteriole, in the smooth muscles that surround that arteriole. And it will respond by constricting the arteriole. So pressure is building up, it's going to constrict it down, and this is going to increase the resistance, which is going to decrease the GFR. So if you think about that hose again, if you squeeze the hose before it gets to the GFR, you're not going to let as much stuff through it. Okay? You're decreasing the amount of stuff that can approach that filter, so you're going to bring your GFR back to baseline. And this is again via that smooth muscle, that vasoconstriction. And this is a negative feedback, so that fall in GFR will then shut off and return the arterial back to its baseline. So changing, vasoconstricting the afferent arterial, we call that myogenic regulation. Increase in mean arterial pressure is the trigger for that. The other thing that happens when you have an increase in that mean arterial pressure, an increase in that MAP, is the macula densa cells respond. Remember, I had shown you that picture of all the complicated stuff right there at the glomerulus. We've got all these different cell types. Well, one of them is in the walls of our distal tubule, which actually it folds back because it's all convoluted and stuff, and hence the name, the distal convoluted tubule. It's all convoluted, so it folds back and it's near the glomerulus. It's there, it's in this crowded region, and it has these cells called macula densa cells. And they can respond to increase in pressure through the distal tubule, and they can also respond to salt concentrations. And what they'll do is they'll send out a chemical signal that will then cause the afferent arterial to constrict. So the end result is the same, that constriction of the arterial, but it can either happen from the stretch receptors in the arterial itself, or it can happen from these macula densa cells nearby. Is this still dealing with myogenic regulation? No, this is tubuloglomerular regulation. This is the second one. Myogenic regulation just means the arterial itself is like, too much, and it gets smaller. This is nearby. It's a good question. So this is the second form of regulation. Tubuloglomerular feedback. So that increase in arterial pressure, that is going to cause an increase in flow through the macula densa. So initially, if we are sending more blood through our afferent arterial, we're going to have a slight increase in GFR, which is bad. We don't want that. But what will happen is that increase in GFR will lead temporarily to an increase in flow through the tubule. And the macula densa cells will be like, we are receiving way too much flow through right now, and they'll send a signal that will be picked up by the afferent arterial, and it will cause it to constrict. So the end result is the same, constricting that afferent arterial, and it can either come from the arterial itself, and/or usually it's both things happening simultaneously from the macula densa cells in the distal tubule. This is the picture of that crowded area that I was talking about, zoomed in. So if this is our whole nephron here, if we focus in around the glomerulus, we have afferent arterial coming up into our glomerulus, our twisted group of blood vessels here, the efferent arterial leaving, and right nearby, we've got this distal tubule here with some macula densa cells, which are specialized cells in the walls of that distal tubule. And the other cells, which we will come back to when we talk more about regulation, are our juxtaglomerular cells, our JG cells right here. So, bunch of cells, afferent arterial can sense increased flow itself, the macula densa cells will sense it. Both of them will work together to decrease the space of the afferent arterial to vasoconstrict so that we can decrease our GFR and keep it constant. Okay, this is the same thing I just said, but shown in like a little flow-through map, if this is the type of information you prefer. Now, I'm the kind of person, I look at this type of visual processing, makes me confused. If you prefer words, ignore this slide. If you're a visual person, see if this makes sense to you. So we talked about those two ways, the myogenic regulation on the left. Increase in mean arterial pressure here in our afferent arterial is going to activate those stretch receptors, which are going to constrict and increase resistance, which will bring down that pressure. And this is a negative, will negatively feedback and shut the whole thing down. However, we also have that increase in arterial pressure that will be picked up by our macula densa cells, which will secrete our paracrine secretion here, is what we call it. So it's going to release a chemical, which will go to the afferent arterial and cause it to constrict, leading to that higher resistance, bringing the pressure down. Okay. The third way is with the mesangial cells. So the mesangial cells are special cells in our nephron that help form the filter of our glomerulus. So the mesangial cells are the cells that form that filter, and the filter itself will change. So a rise in blood pressure will cause the blood vessels to swell, and that will cause stretching of the mesangial cells, those filter cells themselves. And these cells will then start to change the filter in response to that. So it's independent of the vasculature constriction, so it's not going to be changing the afferent arterial. It will just change the amount filtered itself. Okay. The issue with intrinsic regulation, the issue with all of these three ways that we can affect GFR in the face of a change of blood pressure is that they only work over a very narrow range. So if you have a slight increase or a slight decrease in blood pressure, the intrinsic mechanism, your kidney itself, it will take care of it. However, if we have a large change in blood pressure or it happens over a long period of time, think something like chronic dehydration, or you got a lot of blood loss, right? Large decrease in blood volume, that's going to decrease your blood pressure. You need something else. So with that, we're going to use endocrine hormones to try to decrease our GFR, to recover more sodium, and increase our blood volume. Especially when we're talking about blood loss, we want to increase our blood volume. And for this, you want to think about what increases blood volume. Well, it's the recovery of sodium with water following it. So we want to have more water in the blood. That is the volume. More blood volume is more blood pressure. Again, thinking of turning on that hose a little bit more. We're letting more water in. We're increasing the amount, the blood pressure there. And water is going to follow the salt. So we're going to recover more sodium. We will therefore recover more water. Okay, with that goal in mind, let's start to talk a little bit about endocrine hormones. And this brings us to the thing you're diagramming for Friday, our renin, angiotensin, aldosterone system, the RAS system. I typically say RAS system, even though that means like system, but that's just how I've always heard it referred to. But it is our renin, angiotensin, aldosterone system. So I'm going to first go over it. Now, again, I'm trying to appeal to a lot of different learning styles. If I was teaching this in person, I would write things on the board step by step. But I want to make these slides full of words in case people don't watch the video live and they need to learn from words. So I'm going to first go through it in words, and then we're going to go through it in pictures. So if you look at the words and you're like, too many words, relax, pictures are coming. If you look at the pictures and you're like, too many pictures, there are words. Okay, so first in words. Our juxtaglomerular cells, our JG cells, remember they're right there near the glomerulus, near the macula densa cells of the distal tubule. We got the afferent arterial, the efferent arterial, all right together. Those cells are going to release a hormone called renin, with the end goal of increasing our blood pressure. So the goal of the RAS system is to increase blood pressure, and the first molecule involved is renin. Now, what triggers those JG cells are three different things. So the JG cells sense low blood pressure themselves. One, they sense it themselves. They're like, BP is falling, we should release renin. Two, they're also activated by a sympathetic nerve. Now, again, think back to the sympathetic nervous system. This is our fight or flight system. So we want to increase our blood pressure in that fight or flight system. We're going to do that by activating the JG cells to release renin from our sympathetic nerve too. Three, the macula densa cells that we talked about, the macula densa cells in our distal tubule, they sense the low sodium in the filtrate. They sense low sodium in the filtrate, and so they're going to want to signal to the JG cells, hey, we got something going on, we got a situation. Okay. So renin is that's released from our JG cells, is going to activate a molecule called angiotensinogen, which is released from the liver, and it's going to make it angiotensin 1. We're going to start with angiotensinogen. We're going to make angiotensin 1. Angiotensin 1 is then cleaved by a molecule called ACE. It's an enzyme that's released from endothelial cells that line blood vessels. A lot of it is released from endothelial cells in the lungs, but all endothelial cells can release ACE. And ACE will convert angiotensin 1 to angiotensin 2. And what this does is it causes vasoconstriction. So our blood vessels are going to constrict, right? That's going to increase the blood pressure in the same way if you squeeze a hose, Now you're going to be shooting out more at a higher force than if it was relaxed, right? The kidneys are going to reabsorb more salt, and then the pituitary is going to release ADH, and our adrenal glands are going to release aldosterone. Now I know that's a lot. We're gonna unpack this a little bit. Okay, so let's talk about one of the hormones that's the end product of this system, ADH. ADH, antidiuretic hormone. It's also referred to as vasopressin. What it does is it alters the permeability of the collecting duct. So remember, our goal is to increase blood pressure. And one way to do that is through ADH, which is going to say, hey, let's take back some more water directly. And it is the main hormone responsible for the recovery of water from the tubule, from the collecting duct. It's made in the hypothalamus, but released from the pituitary, and it works within minutes to increase the number of aquaporins, our water channels, in the collecting duct. So we're going to take back and recover a lot more water. Okay. One question I want you to think about is the time frame. So ADH is going to work fast. Aldosterone does a similar thing, but takes a lot longer. And I want you to see if you can figure out why that is as we move through their mechanisms. Okay. Keep in mind, osmolarity, water reabsorption. 70% of that happens in the proximal tubule, but this is not regulated. So the amount of water that comes back in the proximal tubule, we cannot control. It's not dependent on our environment. It's not dependent on our needs. However, the rest of the water reabsorption happens in the distal tubule and the collecting duct, and this is where ADH works. So it modulates that 30% of water reabsorption. And how ADH works is, again, ADH, also called vasopressin. It's going to increase the number of aquaporins expressed in the collecting duct. So ADH is going to increase the number of aquaporins expressed in our collecting duct. So if ADH or vasopressin, it binds to a G protein coupled receptor, it's going to work through the adenylene cyclase signaling cascade to make more cyclic AMP. Cyclic AMP will then activate PKA, protein kinase A, which is going to tell, or it's going to phosphorylate specific vesicle proteins that are going to send these vesicles that are already primed and ready to go, already full of aquaporins. It will send them to the surface, and they'll integrate into the cell membrane. And so now we will have very rapidly a lot more aquaporins expressed on the cell membrane and the collecting duct, and we'll be able to take in and reabsorb a lot more water. Okay, so this is the same thing shown another way. So we got ADH, it's flowing through the blood, it's going to bind to that G protein coupled receptor, it's going to work to make more cyclic AMP, which is going to activate protein kinase A, which is going to phosphorylate these vesicles and send all these aquaporins to the surface. And now, all of this water is flowing through. We are taking it back. And what that does is that extra water is going to be taken into our blood vessels. We're going to increase our blood volume, which increases our blood pressure. Remember, blood volume to blood pressure, those are our processes that lead to each other. So increase the water reabsorption, increase the volume of our blood, we increase the pressure. This is showing you how ADH is released, or what controls ADH release. ADH is released by the pituitary. So ADH is made in the hypothalamus, and it's stored and then released from our posterior pituitary when it's been activated by a signal from the hypothalamus. And the hypothalamus can get a few different triggers that tell it to release, to send the signal for ADH release. So angiotensin 2, as we talked about in that RAS system, works by telling the hypothalamus, hey, tell the posterior pituitary, it's time to release ADH. But it also is triggered by the sympathetic nervous system. Again, that fight or flight response, we're going to increase our blood pressure in regards to a fight or flight response. Hyperosmolarity, so if you need more water in your blood, if you got very salty blood, that's going to trigger the hypothalamus. And it's also triggered by decreased atrial receptor firing. So if there is low blood pressure in that regard, the heart can also signal to the hypothalamus, hey, we got a problem. We need to increase our BP. Okay. What triggers that hyperosmolarity specifically in the hypothalamus we have osmoreceptors. So those are the receptors that can get a read on how salty the blood is. Okay, so all of that will trigger the hypothalamus to tell the posterior pituitary to release vasopressin, ADH. Those are the same thing, they're synonyms from each other. And the ADH will tell the blood vessels to constrict, and it will also, again, go to the collecting duct in our nephron, increase aquaporins, increase fluid reabsorption. All of that works together to increase pressure. So if we're increasing the constriction, if we're constricting our vessels, and we're increasing the amount of fluid flowing through that vessel, we're going to increase pressure. Okay, and this is negatively turned off by baroreceptors, which will say, "Hey, we got a high BP. We should turn this whole thing down." Okay, so that is one end product here of this RAS system, is ADH released by the pituitary. Okay. This is the same thing, but shown in another visual, if you like it this way. So if our blood is salty, we got high osmolarity, that's going to tell the hypothalamus, it's going to be detected by those osmoreceptors, hey, it's time to tell the posterior pituitary, release ADH. ADH will not only vasoconstrict, but it will also tell the kidneys, put the aquaporins on the surface. The aquaporins on the surface will increase water reabsorption, turn off down water excretion, and we will have conservation of body water. End result. Higher blood volume, higher BP. We'll then decrease that osmolarity of our extracellular fluid, turning this whole thing off. And again, this is showing you those baroreceptors in the opposite way. If we have low blood volume, again, that whole thing can also be turned on, and that pathway can be activated that way too. So there are a few different activities onto our hypothalamus. Okay, let's talk about the other hormones. So ADH acts quickly. Aldosterone takes a long time, takes a lot longer, several hours, and it regulates sodium and potassium balance. This is our primary mineral corticoid. It's secreted by our adrenal cortex. And what it's going to do is it's going to stimulate the reabsorption of sodium, and water will follow the sodium. It also increases potassium excretion. So it's helping us balance our sodium and potassium levels. And the target here are going to be the principal cells that are in our distal tubule and our collecting duct. So again, we can't regulate what happens at the proximal tubule, but regulation can occur in the end parts of our tubule system, in the distal tubule and the collecting duct. And aldosterone is a steroid hormone. So if you remember back about steroid hormones, they are hydrophobic, so they're going to squeeze on through that plasma membrane like only steroid hormones can do. And they're going to bind to cytoplasmic receptors, and their effect is going to be on transcription. So they have a transcriptional effect. Okay, that was a lot of words. Let's break those words down. Aldosterone, steroid hormone, wiggles through the plasma membrane. It's all hydrophobic. Gets there, activates transcription of some target genes. Activates transcription of target genes for transporters. And these new transporters need to be synthesized and folded and put into vesicles and brought to membranes. And its gene targets are things like the sodium-potassium pump in the basolateral membrane of our principal cells, as well as potassium and sodium channels on that apical membrane. And what happens is that there is a net exchange of plasma potassium for sodium in the urine. So we're going to reabsorb more sodium and we're going to excrete more potassium. And there's no direct effect on water, but water will follow the sodium. Water will follow the sodium. This is what that looks like again. So aldosterone, it is going to wiggle on through. It's going to go to its cytosolic receptor. It's going to turn on these genes that are going to make more of those sodium potassium ATPase, those pumps that are going to put more sodium into the blood and more potassium into the filtrate. It's also going to make more channels so that when we build and create these gradients, that those ions can then flow. So if we create a gradient where there's not a lot of sodium inside the epithelial cell, we want to have these channels, these extra channels, so that more sodium can flow from the filtrate down its gradient into the epithelial cell, where it will quickly be pumped into the interstitial, the peritubular, fluid here, and go into the blood. Okay. So we talked about what happens again to fall when we have falling blood pressure, how our body wants to increase that BP. I want you to pay attention to what hormones target and what regulates them. We'll continue this discussion. We started talking today about the RAS system. I want you, after this class, to work on drawing out those pieces. We're going to keep expanding upon that on Friday, so we'll add more to your drawing, but I want you to really take a minute and see if you can talk through what happens and where those specific hormones are working. Okay. Let me play with technology here a second. Okay. So that's where we're going to stop for today. On Friday, we are going to finish up the RAS system. Again, I know it's a lot of complicated material. I really want you to take a few minutes and draw it out. And then if you're still confused, Khan Academy has some good videos about the RAS system that I posted on the YouTube video page that will help you as well. Okay, so that is it for class today. I'll stick around if you guys have questions. Otherwise, I will see you Friday.